So hello dear learners, in this video we are going to study chapter 15 body fluids and circulation of class 11th NCERT biology. In this chapter we will cover these six topics and these are first blood, second is lymph tissue fluid, third circulatory pathways, double circulation, regulation of cardiac activity, disorders of circulatory system. So here we will cover these topics. Here we start. You have learned that living cells have to be provided with nutrients, O2 and other essential substances. Also, the waste or harmful substances produced have to be removed continuously for healthy functioning of tissues. It is therefore essential to have efficient mechanisms for the movement of these substances to the cells and from the cells. Different groups of animals have evolved different methods for this transport. Simple organisms like sponges, ants, sealant threads circulate water from their surroundings through their body cavities to facilitate the cells to exchange these substances. More complex organisms use special fluids within their bodies to transport such materials. Blood is most commonly used body fluid by most of the higher organisms, including humans. For this purpose, another body fluid, lymph, also helps in the transport of certain substances. In this chapter, you will learn about the composition of and properties of blood and lymph tissue fluid, and the mechanism of circulation of blood is also explained herein. So the first heading is blood. Blood is a special connective tissue consisting of a fluid matrix, plasma, and formed elements. Next is plasma. Plasma is a straw-colored viscous fluid consisting nearly 55% of the blood. 90 to 92% of plasma is water, and proteins contribute 6 to 8% of it. Fibrinogen, globulins, and albumins are the major proteins. Fibrinogens are needed for the clotting and coagulation of blood. Globulins primarily are involved in defense mechanism of the body, and albumins help in osmotic balance. Plasma also contains some small amount of minerals like Na plus, Ca plus plus, Mg plus plus, HCO minus, HCO3 minus, Cl minus, etc. Glucose, amino acids, lipids, etc. are also present in the plasma as they are always in transit in the body. Factors for coagulation for clotting of the blood are also present in the plasma in an inactive form. Plasma without clotting factors is called serum. Formed elements. Erythrocytes, leukocytes, and platelets are collectively called formed elements, and they constitute nearly 45% of the blood. Erythrocytes are red blood cells, are the most abundant of all cells blood. A healthy adult man has on an average 5 millions to 5.5 millions of RBCs milli per meter milli per meter of blood. RBCs are formed in the red bone marrow in the adults. RBCs are devoid of nucleus in the most mammals and are biconcave in the shape. They have a red colored iron containing complex protein called hemoglobin, hence the color and the name of these cells. A healthy individual has 12 to 6 grams of hemoglobin in every 100 ml of blood. These molecules play a significant role in transport respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed and spleen graveyard of RBCs. Next, leukocytes are also known as white blood cells as they colorless due to the lack of hemoglobin. They are nucleated and are relatively lesser in number, which average 6,000 to 8,000 milli per meter cube of blood. Leukocytes are generally short-lived. We have to maintain two main categories of WBCs, granulocytes and agranulocytes. Neutrophils, eosinophils, and basophils are different types of granulocytes, while lymphocytes are and monocytes are agranulocytes. Neutrophils are the most abundant cells, 60 to 65% of the total WBC, and basophils are least 0.05 to 1%. among them. Neutrophils are monocytes, 68% are phagocytic cells which destroy foreign organism entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reactions. Eosinophils 2 to 3% resist infections and are also as- associated with allergic reactions. Lymphocytes 20 to 25% are of the two major types B and T forms. Both B and T lymphocytes are responsible for immune responses for the body. Platelets are also called thrombocytes are cell fragments produced from megakaryocytes, special cells in bone marrow. Blood normally contains 1,50,000 to 3,50,000 platelets per meter milli per meter. Platelets can release a variety of substances, most of which are involved in coagulation for or clotting of blood. A reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. Next is blood groups. As you know, blood of human beings differ in certain aspects, though it is appears to be similar. Various type of grouping of blood has been done. Two such groupings, ABO and RH, are widely used all over the world. ABO grouping. ABO grouping is based in the presence or absence of two surface antigens chemicals that can induce immune response on the RBCs, namely A and B. Similarly, the plasma of different individuals contain two natural antibodies. The distribution of antigens and antibodies in the four groups of blood A, B, and O are given in table 15.1. You probably know that during blood transfusion, any blood cannot be used. The blood of a donor has to be carefully matched with the blood of the recipient before any blood transfusion to avoid severe problems of clumping. The donors of compatibility is also shown in table 15.1. So, blood group A, antigens on RBCs A, antibodies in plasma NTB. Donors group is A and O. So, A can donate its blood and O can also donate its blood. B, antigens on RBC B, antibodies in plasma NTA. Donors are B and O. So next is AB. Antigens on RBCs A and B, antibodies in plasma nil. Donors group AB, A, B, O. Blood group O, antigens on RBC nil, antibodies in plasma anti A and B. Donors group O. So O can donate its blood to all A, B, and AB. From the above mentioned table, it is evident that group O blood can be donated to persons with any other blood group, and hence O group individuals are called universal donors. Person with AB group can accept blood from persons with AB as well as the other group of blood. Therefore, such persons are called universal recipients. RH grouping. Another antigen, RH antigen, similar to one present in rhesus monkeys, is also observed in surface of RBCs of majority, nearly 80% of humans. Such individuals are called RH positive, and those in whom antigen is absent is called RH negative. And positive RH negative person is, if exposed to RH positive blood, will form specific antibodies against RH antigens. Therefore, RH group should be matched before transfusions. A special case of RH incompatibility has been observed between RH negative blood of a pregnant mother with RH positive blood of the fetus. RH antigens of the fetus do not get exposed to the RH negative blood of the mother in first pregnancy, as two bloods are well separated by the placenta. However, during the delivery of the first child, there is a possibility of exposure of the maternal blood to small amounts of RH positive blood from the fetus. In such cases, the mother starts preparing antibodies against RH antigen in her blood. In case of her subsequent pregnancies, the RH antibodies from the mother, RH negative, can leak into the blood of the fetus and destroy the fetal RBCs. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis foetalis. This can be avoided by administering anti-RH antibodies to the mother immediately after the delivery of the first child. Next is coagulation. Next is coagulation of blood. Next is coagulation of blood. You know that when you cut your finger or hurt yourself, your round does not continue to be bleed for a long time. Usually the blood stops flowing after some time. Do you know why? Blood exhibit coagulation or clotting in response to an injury or trauma. This is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish brown scum formed at a site of cut or an injury or a period of time. It is a clot or coagulum formed mainly of a network of threads called fibrins, in which dead and damaged formed elements of blood are trapped. Fibrins are formed by the conversion of inactive fibrinogens and the plasma by enzyme thrombin. Thrombins in turn are formed from another inactive substance present in the plasma called prothrombin. An enzyme complex thrombokinase is required for the reaction. This complex is formed by a series of linked enzymic reactions, cascade process involving a number of factors present in the plasma. In an active state, an injury or a trauma stimulates the platelets in the blood to release certain factors which activate the mechanism of coagulation. Certain factors released by the tissues at the site of injury also can initiate coagulation. Calcium ions play a very important role in clotting. Next is lymph tissue fluid. As the blood passes through the capillaries in the tissue, some water along with many small water soluble substances move out in the spaces between the cells of the tissues, leaving the larger proteins in the and the moist of the former elements in the blood vessels. This fluid released out is called interstitial fluid or tissue fluid. It has the same mineral distribution as that in plasma. Exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. An elaborate network of vessels called lymphatic system collects this fluid and drains it back to the major veins. The fluid present in the lymphatic system is called the lymph. The lymph is colorless fluid containing specialized lymphocytes which are responsible for the immune responses of the body. Lymph is also an important carrier for nutrients, hormones, etc. Fats are absorbed through lymph in the lacteals present in the intestinal villi. Circulatory pathways. The circulatory patterns of the two types, open or closed. Open circulatory system is present in arthropods and mollusks, in which blood pumped by the heart passes through the large vessels into open spaces or body cavities called sinuses. Annelids and chordates have closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of blood vessels. This pattern is considered to be more advantageous as the flow of fluid can be more precisely regulated. All vertebrates possess a muscular chambered heart. Fishes have two chambered heart with an atrium and a ventricle. Amphibians and the reptiles, except crocodiles have three chambered heart with two atria and single ventricle. Whereas crocodiles, birds, and mammals possess a four-chambered heart with two arteries and two ventricles. In fishes, the heart pumps out deoxygenated blood, which is oxygenated by the gills and supplied to the body parts, from where deoxygenated blood is returned to the heart. In amphibians and reptiles, the left atrium receives oxygenated blood from the gills or lungs or skin, and the right atrium gets deoxygenated blood from other body parts. However, they get mixed up in single ventricle, which pumps out mixed blood. Incomplete double circulation in birds and mammals. Oxygenated and deoxygenated blood received by left and right atria respectively passes to the ventricles of the same sides. The ventricles pump it out without any mixing up. It means two separate circulatory pathways are present in these organisms. Hence, these animals have double circulation. Let us study the human circulatory system. Human circulatory system. Human circulatory system, also called the blood vascular system, consists of a muscular chambered heart, a network of close branching blood vessels, and blood, the fluid which is circulated. Heart. The mesodermally derived organ is situated in the thoracic cavity in between the two lungs, slightly tilted to the left. It has size of clenched fist. It is protected by double-walled membranous bag Pericardium, enclosing the pericardial fluid. Our heart has four chambers, two relatively small upper chambers called atria, and two larger lower chambers called ventricles. A thin muscular wall called the interatrial septum separates the right and the left atria, whereas a thick wall, the interventricular septum, separates the left and right ventricles. The atrium and the ventricle of the same side are also separated by a thick fibrous tissue called the atrioventricular septum. However, each of these septa are provided with an opening through which the two chambers of the same side are connected. The opening between the right atrium and the right ventricle is guarded by a wall formed of three muscular flaps or cusps. The tricuspid valve, whereas bicuspid or mitral valve guards the opening between the left atrium and the left ventricle. The opening of the right and the left ventricles into the pulmonary artery and the aorta respectively are provided with the semilunar valves. The valves in the heart allows the flow of blood only in one direction. It means from the atria to the ventricles and from the ventricles to the pulmonary artery or aorta. These valves prevent any backward flow. The entire heart is made up of cardiac muscles. The walls of ventricles are much thicker than that of atria. A specialized cardiac musculature called the nodal tissue is also distributed in the heart. A patch of this tissue is present in the right upper corner of the right atrium called sinoatrial node (SAN). Another mass of this tissue is seen in the lower left corner of the right atrium close to the atrioventricular septum called the atrioventricular node (AVN). A bundle of nodal fibers, atrioventricular bundle (AV bundle), continues from the AVN, which passes through the atrioventricular septa to emerge on the top of the interventricular septum and immediately divides into a right and left bundle. These branches give rise to minute fibers throughout the ventricular musculature of the respective sites and are called Purkinje fibers. The nodal musculature has the ability to generate action potentials without any external stimuli. It is auto-excitable. However, the number of actions potential that could be generated in a minute vary at different parts of the nodal system. The SAN can generate the maximum number of action potentials. It means 70 to 75 per minute and is responsible for the initiating and maintaining the rhythmic contractile activity of the heart. Therefore, it is called pacemaker. A heart normally beats 70 to 75 times in a minute, average 72 beats per minute. Cardiac cycle. How does the heart function? Let us take a look. To begin with, all the four chambers of the heart are in a relaxed state. It means they are in a joint diastole. As the tricuspid and bicuspid valves are open, the blood from the pulmonary veins or vena cava flows into the left and right ventricle respectively to the left and right atria. The semilunar valves are closed at this stage. The SAN now generates an action potential which stimulates both the atria to undergo a simultaneous contraction. The atrial systole. This increases the flow of blood into the ventricles by about 30%. The action potential is conducted to the ventricular side by AVN and AV bundle, from where the blood bundle office transmits it to the entire ventricular musculature. This causes the ventricular muscles to contract. The atria undergoes relaxation diastole, the coinciding with the ventricular systole. Ventricular systole increases the ventricular pressure, causing the closure of tricuspid and bicuspid valves due to attempted backflow of blood into the atria. As the ventricular pressure increases further, the semilunar valves guarding the pulmonary artery and the aorta left side are forced open, allowing the blood in the ventricles to flow through these vessels into the circulatory pathways. The ventricles now relax and the ventricular pressure falls, causing the closure of semilunar valves, which prevents the backflow of blood into the ventricles. As the ventricular pressure declines further, the tricuspid and bicuspid walls are pushed open by the pressure in the atria exerted by the blood, which was being emptied into them by the veins. The blood now once again moves freely to the ventricles. The ventricles and atria are now again in a relaxed state as earlier. Soon SAN generates a new action potential in the events described above are repeated in that sequence and the process continues. This sequential events in the heart, which is cyclically repeated, is called cardiac cycle, and it consisted of systole and diastole of both the atria and ventricles. As mentioned earlier, the heart beats 72 times per minute. It means that many cardiac cycles are performed per minute. From this, it could be deduced that the duration of cardiac cycle is 0.8 seconds. During a cardiac cycle, each ventricle pumps out approximately 70 ml of blood, which is called stroke volume. The stroke volume multiplied by the heart rate number of beats per minute gives the cardiac output. Therefore, the cardiac output can be defined as the volume of blood pumped out by each ventricle per minute and average 5000 ml of 5 liters in a healthy individual. The body has the ability to enter the stroke volume as well as the heart rate and thereby the cardiac output. For example, a cardiac output of an athlete will be much higher than that of an ordinary man. During each cardiac cycle, two prominent sounds are produced which can be easily heard through a stethoscope. The first heard sound LUB is associated with the closure of tricuspid and bicuspid valves, whereas the second heard sound lub and dub is associated with closure of the seminal valves. These sounds are of clinical diagnostic significance. And next is electrocardium. Next is electrocardiogram. You are probably familiar with this scene from a typical hospital television show. A patient is hooked up to a monitoring machine that shows voltage traces on a screen and makes the sound pip 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 pee as the patient goes cardiac arrest. This type of machine electrocardiograph is used to obtain an electrocardiogram ECG. ECG is a graphical representation of electrical activity of the heart during a cardiac cycle. To obtain a standard ECG as shown in the figure 515.3, a patient is connected to the machine with three electrical leads that continuously monitor the heart activity. For a detailed evaluation of the heart's function, the multiple leads are attached to the chest region. Here, we will talk only about a standard ECG. Each peak in the ECG is identified with a letter form P to T that corresponds to a specific electrical activity of the heart. The P wave represents the electrical excitation or depolarization of the atria, which leads to contraction of both the atria. The QRS complex represents depolarization of the ventricles, which initiates the ventricular contraction. The contraction starts shortly after Q and marks the beginning of the systole. The T wave represents the return of the ventricles from excited to normal state, and the end of T wave marks the end of systole. Obviously, by counting the number of QRS complexes that occur in a given time period, one can determine the heartbeat rate of an individual. Since the ECGs obtained from different individuals have roughly the same shape for a given lead configuration, any deviation from this shape indicates a possible abnormality or disease. Hence, it is of a great clinical significance. Double circulation. The blood flows strictly by a fixed route through blood vessels. The arteries and veins. Basically, each artery and vein consists of three layers: an inner lining of squamous endothelium and tunica intima, a middle layer of smooth muscle and elastic fibers, the tunica media, and an external layer of fibrous connective tissue with collagen fibers, the tunica externa. The tunica media is comparatively thin in the veins. Figure 15.4. As mentioned earlier, the blood
As mentioned earlier, the blood pumped by the right ventricle enters at the pulmonary artery, whereas the left ventricle pumps blood into the aorta. The deoxygenated blood pumped into the pulmonary artery is passed on to the lungs, from where the oxygenated blood is carried by the pulmonary veins into the left atrium. This pathway constitutes the pulmonary circulation. The oxygenated blood entering the aorta is carried by a network of arteries, arterioles and capillaries to the tissues, from where the deoxygenated blood is collected by a system of veins, and vena cava and emptied into the right atrium. This is the systemic circulation. The systemic circulation provides nutrients, O2 and other essential substances to the tissues, and takes CO2 and other harmful substances away for elimination. A unique vascular connection exists between the digestive tract and liver, called hepatic portal system. The hepatic portal vein carries blood from intestine and to the liver before it is delivered to the systemic circulation. A special coronary system of blood vessel is present in our body exclusively for the circulation of blood to and from the cardiac musculature. Regulation of cardiac activity. Normal activities of heart are regulated intrinsically. It means auto-regulated by specialized muscles, nodal tissue. Hence, the heart is called myogenic. A special neural center in the medulla oblongata can moderate the cardiac function through autonomic nervous system ANS. Neural signals through the sympathetic nerves part of ANS can increase the rate of heartbeat, the strength of ventricular contraction, and thereby the cardiac output. On the other hand, parasympathetic neural signals, another component of ANS, decrease the rate of heartbeat, speed of conduction, and action potential, and thereby the cardiac output. Adrenal medullary hormones can also increase the cardiac output. Disorders of circulatory system. High blood pressure. Hypertension. Hypertension is a term for blood pressure that is higher than normal. In this measurement, 120 mmHg millimeters of mercury pressure is a systolic or pumping pressure, and 80 mmHg is a diastolic or resting pressure. If repeated checks of blood pressure of an individual is 140/90 or higher, it shows hypertension. High blood pressure led to heart diseases and also affect vital organs like brain and kidney. Coronary artery disease, CAD. Coronary artery disease, often referred to as atherosclerosis, affect the vessel that supply blood to the heart muscle. It is caused by deposits of calcium, fat, cholesterol, and fibrous tissue, which make the lumen of arteries narrower. Angina. It is called angina pectoris. Angina pectoris. A symptom of acute chest pain appears when no enough oxygen is reaching the heart muscle. Angina can occur in men and women of any age, but it is more common among the middle-aged and elderly. It occurs due to conditions that affect the blood flow. Heart failure. Heart failure means the state of heart when it is not pumping blood effectively enough to meet the needs of the body. It is sometimes called congestive heart failure because congestion of the lung is one of the main symptoms of this disease. Heart failure is not same as cardiac arrest when the heart stops beating, or a heart attack when a heart muscle is suddenly damaged by an inadequate blood supply. Summary. Vertebrates circulate blood, a fluid connective tissue in their body, to transport essential substances to the cells and to carry waste substances from there. Another fluid, lymph tissue fluid, is also used for the transport of certain substances. Blood comprises of fluid matrix, plasma, and formed elements. Red blood cell RBCs, erythrocytes, white blood cell WBCs, leukocytes and platelet thrombocytes constitute the formed element. Blood of humans are grouped into AB and O system based on the presence and absence of two surface antigens A and B on the RBCs. Another blood grouping is also done based on the presence or absence of another antigen called rhesus factor Rh on the surface of RBCs. The spaces between cells in the tissues contain a fluid derived from blood called tissue fluid. This fluid is called lymph, is almost similar to blood except for the protein content and the formed element. All vertebrates and a few invertebrates have a closed circulatory system. In our circulatory system consists of a muscular pumping organ, a heart, a network of vessels, and a fluid blood. Heart has two atria and two ventricles. Cardiac musculature is auto-excitable. Sinoatrial node SAN generates the maximum number of action potential per minute, 70 to 75 per minute, and therefore it sets the pace of activities of heart. Hence it is called pacemaker. The action potential causes the atria and then the ventricle to undergo contraction followed by their relaxation. The systole forces the blood to move from the atria to the ventricles and to the pulmonary artery to the and the aorta. The cardiac cycle is formed by the sequential events in the heart, which is cyclically repeated and is called cardiac cycle. A healthy person's 72 such cycle per minute, about 70 ml of blood is pumped out each ventricle during a cardiac cycle, and it is called the stroke or beat volume. Volume of blood pumped out by each ventricle of heart per minute is called the cardiac output, and it is equal to the product of stroke volume and heart rate. The electrical activity of heart can be recorded from the body surface of by using electrocardiograph, and recording is called ECG, which is of clinical importance. We have a complete double circulation. It means two circulatory pathways, namely pulmonary and systemic are present. The pulmonary circulation starts by the pumping of deoxygenated blood by the right ventricle, which is carried to the lungs where it is oxygenated and returned to the left atrium. The systemic circulation starts with the pumping of oxygenated blood by the left ventricle to the aorta, which is carried to all the body tissues, and deoxygenated blood from there is collected by the veins and returned to the right atrium. Though the heart is auto-excitable, its function can be moderated by neural and hormonal mechanisms. So our chapter is finished. Thanks for watching.